proteins are digested into amino acids, dipeptides, and tripeptides that can then be absorbed in the small intestine. So protein sources in the small intestine include both ingested protein in the diet and, in addition, a large amount of endogenous protein. And that comes from the enzymes and mucus that are secreted into the lumen, and especially those epithelial cells that are apoptosed and that are constantly being sloughed off into the lumen during the renewal process. Protein is a polymer of amino acids, and it's bound by peptide bonds. And this protein is required to supply essential amino acids and to replace the nitrogen contained in amino acids that are metabolized to urea. The proteolytic enzymes, sometimes called proteases or peptidases, break peptide bonds within a protein to get to these dipeptides, tripeptides, and free amino acids that can be absorbed in the small intestine. So the proteolytic enzymes are stored in the body as these inactive zymogens, and the reason that this is done is to protect the secreting organ from autodigestion by the enzyme. So the enzymes are then activated once they reach the lumen where they're going to be performing their digestive function. And these proteolytic enzymes can be either endopeptidases that cleave the internal peptide bonds between amino acids, resulting in oligopeptides, or exopeptidases that break peptide bonds on the ends of the molecule, creating a free amino acid. This is just an overview of the components important in protein digestion. So protein digestion begins in the stomach with digestion by pepsin. It then continues in the small intestine with secretions of pancreatic proteases, trypsin, chymotrypsin, carboxypeptidase, and elastase, until finally small peptides are digested by brush border peptidases, the amino oligopeptidase and dipeptidases, for example, that break them down into the absorbable amino acids, dipeptides, and tripeptides. So protein digestion initially occurs in the gastric lumen by the enzyme pepsin. So we said that these chief cells secrete the inactive zymogen pepsinogen, and that's secreted into the lumen, and then in the presence of acid in the gastric lumen, that acid secreted by the parietal cells, the pepsinogen is activated, undergoing autocatalytic cleavage to become the active pepsin. So pepsinogen in the presence of acid cleaves itself, becoming pepsin, and that pepsin is an enteropeptidase that preferentially cleaves proteins at neutral amino acids and converts large peptides into smaller oligopeptides. So pepsin's only active at acidic pH, and it's inactivated at a pH above about 4.5. So this means that these gastric pepsins are quickly inactivated once they reach the small intestine as a result of the bicarbonate coming from both the Brunner's glands and the duodenal epithelium, as well as the pancreatic ducts and the biliary system. Pepsin isn't absolutely necessary for normal protein digestion, so even in the absence of pepsin, protein is still fully digested in the small intestine by the pancreatic enzymes and brush border enzymes. The majority of proteolysis occurs in the lumen of the small intestine, as we'll see here. So once peptides move from the stomach and into the duodenum, they're further digested by pancreatic proteases. So the exocrine pancreas secretes these 5 inactive enzymes or zymogens that then become activated once they reach the small intestine. So one of those enzymes secreted in zymogen form by the exocrine pancreas is trypsinogen. And trypsinogen is initially activated by the brush border enzyme enterokinase to yield the endopeptidase, the active trypsin. And we can kind of see that here where this is the brush border enzyme endopeptidase. So when trypsinogen that is released from the pancreas interacts with endopeptidase, it becomes this active enzyme trypsin. trypsin is then able to autoactivate itself, forming more trypsin, but it also activates the other zymogens. So trypsin is responsible for not only creating more trypsin from trypsinogen, but it's also responsible for creating the active chymotrypsin from inactive chymotrypsinogen, creating elastase from proelastase, and creating these active carboxypeptidases from the procarboxypeptidases. And as you can see here, the trypsin, chymotrypsin, and elastase are all endopeptidases, while the carboxypeptidases are exopeptidases, which cleave that end amino acid off, resulting in release of a free amino acid. After digestion by that pepsin in the stomach and by the pancreatic proteases in the intestine, there are many small peptides present in the intestinal lumen that undergo further breakdown at the brush border. So there are multiple different peptidases present on the brush border of the enterocytes, some of them endopeptidases, some of them exopeptidases, along with that specific enterokinase that's important in catalyzing the activation of trypsinogen to trypsin. So each of these peptidases recognizes a limited number of peptide bonds, and that's why it's important to have many different peptidases present to ensure the complete breakdown of peptides in the intestinal lumen. These brush border peptidases are what complete protein digestion of these small peptides, which are what are primarily present at this point, into the absorbable components. And the final products are these di and tripeptides and free amino acids, both of which can be absorbed by the enterocyte. So as we saw, amino acids as well as di and tripeptides can all be absorbed from the lumen by the enterocyte using a carrier-mediated process. So free amino acids are transported across the luminal membrane by a sodium amino acid cotransporter, and this is a secondary active transport process like we've seen many times before, and it's of course driven by the sodium gradient that's maintained by this basolateral sodium-potassium ATPase. The dipeptides and tripeptides are transported across the luminal membrane by a hydrogen ion peptide cotransporter, and these have a pretty broad specificity, and this is called a PEP-T1 transporter, and this is a form of secondary active transport coupled to hydrogen ions. So the hydrogen ion gradient is maintained by a sodium hydrogen exchanger that is also in the luminal membrane, although not pictured in this image. Then once inside the enterocyte, most dipeptides and tripeptides are further broken down by some cytoplasmic peptidases into free amino acids. And then all of the free amino acids can be transported across the basolateral membrane through multiple transporters by facilitated diffusion. There may be a few remaining di and tripeptides that can potentially be absorbed unchanged as well.